Assalamualaikum. We are from Gutsy and we are going to present about egg-drop syndrome. Egg-drop syndrome 76, also known as EDS 76, is a disease that caused by a adenovirus. The effects are the incubation period is 7 until 9 days, loss of colors in pigmented egg, thin shell, soft shell or shell-less egg, a reduction in production up to 40%, watery albumin and reduction in egg size. The egg with dullness and diarrhea may occur. Typically, the disease in hand is known as egg-drop syndrome, but the full name is egg-drop syndrome 76 or EDS 76. However, we should use the full name to make it easier to distinguish it from recently known dark egg disease that called egg-drop syndrome in dark or dark egg syndrome. Different diseases for different species should be applied so as not to give rise to potential confusion. Egg-drop syndrome 76 attacks chicken and has been occurred in several countries such as Uruguay, England, Germany, Spain, Holland, France, Peru, Brazil, Ireland, and Argentina. How to test freshness of the egg, whether it is good or not. There are 3 easy ways to determine and test the freshness of the egg, which is the sniff test, the egg white test, and the floats and sing test. For the sniff test, normally when we smell sulfur, we will describe it as a rotten egg. Due to a baked egg, we emit a strong sulfur odor. If the egg is really past prime time, it is likely that the bad smell of the egg can be inhaled through the shell. But if it does not have any odor and you are worried about freshness, then be careful after you break it. For the egg white test, first you need to crack the egg onto a plate or other flat surface. Look closely at the consistency of the egg white, and it should be slightly opaque, not spread out too much, and appear thick and a bit sticky. If it is watery, clear, and runny, the egg has lost its freshness. This is due to the fact as egg age, the white turn liquidy and break down, and the yolk will be slightly flat on top instead of round. Lastly, for the floats and sing test, all you need is egg, a bowl, and cool water. First, you need to fill the bowl with enough cool water to completely cover the egg. Then slowly drop the egg into the bowl of water. If it sinks to the bottom, turn on its sides and stay there. It is very fresh. If the egg sinks but floats at an angle or stand on end, the egg is a bit older, maybe a week to 2 weeks old, but it is still okay to eat. If the egg floats, then the egg is too old and you should discard it.
Okay, now we move to the mode of transfer, which means how the virus spread or transfer to other place and infect others. There are 2 types of transmission, which is the vertical and horizontal transmission. The vertical transmission is the passage of disease-causing agent from mother to their baby. And as for the chicken, it means that from the hen to their eggs. While horizontal transmission means the spread of infectious agent from one individual to another, usually through contact with bodily excretion or fluid, such as sputum or blood that contain the agent. The contamination of egg trays also played a part in transmission as white foam, their dropping and biting insect. However, for the biting insect is not proven yet. For etiology, EDS virus is caused by duck adenovirus A and was first described in laying hen in 1976. The adenovirus is a member of genus Atadenovirus and family Adenoviridae. The natural host for this virus is wild and domestic waterfowl such as duck and geese, but they are asymptomatic carrier, and it start to spread to chicken through contaminated vaccine and contaminated drinking water by dropping. And to diagnose this disease, it's not enough to just look in terms of history, sign or lesion in chicken because it is hard to identify because chicken often look healthy. Therefore, the best way is to do laboratory tests, which is the serological testing. The serology testing is an antibody test that uh, that look for the presence of antibodies, which is a specific protein in a specific protein made in response to infection. The common tests of choice include hemagglutination inhibition, ELISA, and serum neutralization. This testing can determine if chicken have an antibodies against EDS, which would indicate that they have been infected. This method is only suitable for chicken that have not been vaccinated. And another method that can be used is by feed uh, egg with EDS to unvaccinated chicken and identify of the antibodies in the chicken.
Next is postmortem of the disease. How does an hen produce egg? So the egg is formed in the reproductive tract of a female chicken, which is called as hen, and reproductive tract is divided into two major parts, which is ovary and oviduct. As you can see in the pictures, this one is the ovary part where the yolk is formed. After it reaches to the right size, it will be released from the ovary by the process called ovulation. Then the released yolk is picked up by infundibulum, and hence the fertilization will take place. Fertilization will take place. Then the yolk will pass through the magnum where the albumin will form, and then it will go to the isthmus for additional of cell membrane, and then it will go to the shell gland of uterus where the shell will be formed like fully, something like this, full shell, and then it will pass through the vagina. So the vagina is push out the larger part of larger end first, which is example of this is the larger end of an egg, and after the hen is drop out of an egg, this one will be the upper part and this one will be the lower part. So this prevents the egg from being contaminated by fecal material when it is laid. Process of egg drop syndrome reproductive part, which is looks something like this, affected chicken reproductive parts. So usually these are not a naturally occurring infection due to the inactiveness of ovary and oviduct atrophy, which is meaning lack of cell, organ and tissues due to the lack of nutrition or not a good type of feeds which is provided to the hen, and so on. So uterine edema has been reported in a previous outbreak. Other lesions are not discovered yet. Hence, the replicant of the virus in epithelial cells of oviduct glands result in several inflammatory and drysophic changes in the mucus coat. So, to compare the healthy and unhealthy egg, which is egg drop syndrome, it's like the egg contains enough of water, protein, fat, carbohydrate and hash, and physical composition of an healthy egg is shell is 10.3%, albumin inside of it is 57.6%, and the yolk is 32.1%, and if it contains all this kind of composition, that is considered as an healthy egg. So the egg drop syndrome happens because of lack of nutrition, and so the produced egg will be in bad condition. That's it. So, for the example, this one is the egg drop syndrome example, and this one is the healthy egg.
So here I'm going to explain about the sign or symptom of egg drop syndrome. As we all know, egg drop syndrome is a viral disease that results in decreased egg production. Flocks normally won't show any symptoms of having egg drop syndrome until it starts to lay eggs. So, in flux without antibody, the first sign of the disease is the production of pale shell eggs, quickly followed by the production of thin or soft shell eggs, or we also call it as shellless eggs. Another signs are loss of shell pigment and also poor internal quality. So here are some pictures of egg drop syndrome. The first picture starting from the right showed an egg without the shell, which we call it as shellless egg. The next picture showed a picture of pale shell eggs, and the last picture we have here showed an egg with a soft shell. So the thin or soft shell eggs and shellless eggs are very fragile, so the birds tend to eat them. And these eggs also get trampled into the litter and may be overlooked unless a careful examination is made.
Prevention and treatment. Actually, there is no treatment for egg drop syndrome 76. The classic form has been eradicated from primary breeders. Use of dedicated equipment and egg trays for each farm or washing and disinfecting plastic egg trays before use can help to control the endemic form. The sporadic form can be prevented by separating chickens from other birds, especially waterfowl. General sanitary precautions are indicated and potentially contaminated water should be chlorinated before use.
So, as a conclusion for suggestion, farmers need to first use dedicated equipment and egg tray for each farm. Second, washing and disinfecting plastic egg trays before use. Third, separate chickens from other birds, especially waterfowl. Fourth, the last one is contaminated water should be chlorinated before use. In our opinion, even though there is no treatment, but the vaccines are available to prevent this infection for egg drop syndrome. If we appropriately produce and administer this inactive vaccine can prevent and reduce egg drop syndrome disease, even it can't prevent this viral shedding. So farmers need to be well aware to vaccinate hen during growing period around 14 to 18 weeks age. Okay, that's all from us. Thank you.